To summarize what we've talked about, the main activated pathways we looked at were CD163 mediating an anti-inflammatory response, transfer of LPS from LBP carriers to CD14, neurophilin interactions with VEGF and VEGFR, the aim to inflammasome. These pathways were all enhanced and upregulated, so it could be interesting to try and downregulate these genes, the genes that are associated with them, and see what effect it would cause specifically on glioblastoma cells. We also analyzed 3 highly expressed genes in glioblastoma patients, and these were HCK, MCM8, and EME1. These genes have already been studied, but not to a large extent. And nevertheless, results are quite promising for them, as knocking them down and reducing the number of copies they were present in caused significant changes, as demonstrated by how much longer xenografts or patients ended up living. Our suggested targets subsequently are these 4 pathways and 3 genes. However, more research should be conducted with cell lines or xenografts before targeting them in humans with glioblastoma. MCM8 and EME1 both don't have drugs against them at all, while HCK has been targeted, but only non-selectively. A possible experimental direction could be targeting these genes selectively, the HCK gene. So I looked for possible overlaps between the studied genes and pathways and actually found some interesting information. I didn't investigate them deeply, but there was an article about CD163 and CD14 interaction, for example, while another one talked about VEGF being involved in the AIM2 inflammasome pathway. But more research could be conducted to potentially combine targeted therapy of the different genes and pathways discussed. Thank you.